So today we're going to start talking about a process called photosynthesis, something that pretty much all of life on Earth depends on. It is the basic chemical process that allows for the sunlight to be converted to a usable form of energy for all living organisms. But first I want to just step back and kind of put it into a little bit of a context. So there are two main energy converting organelles within cells. There's the chloroplast and the mitochondria. Clearly the chloroplast is only going to be found in plant cells, and mitochondria are found in both plant and animal cells. But both of them play a role in converting different types of energy into a usable form for the cells. And do you remember what that usable form of energy is? ATP. Very good. So in order for energy to be produced in the form of ATP, both chloroplasts and mitochondria use what's called chemiosmotic coupling. Now the basic definition of that is going to be the common pathway by which mitochondria and chloroplasts harness energy for biological purposes of the cell. So what energy are we harnessing? In the chloroplast, we're going to harness sunlight energy, and in mitochondria, we're going to harness the energy that's stored within food. So as we do that, the energy from either the sunlight or the oxidation of foodstuffs is going to be used to drive membrane-bound proton pumps. And you can see that in this image right here. We have three of them. They're blue. And those hydrogen pumps are going to generate what we call an electrochemical proton gradient across this membrane. So electrochemical gradient being that we've taken all these hydrogen ions and pumped them onto one side of the membrane. So what we have then is what we call a high potential energy, particularly on this side of the membrane. And note that these hydrogen pumps are driven by this electron transport chain, is what this is called. It's where electrons are passed from one compound to another. And we're going to talk a little bit about that in a minute. But let's look at our overall picture. If we have a high potential energy because of this now huge concentration gradient of hydrogen ions, we can then use that as we release that potential energy and allow it to be converted to chemical energy. We then allow the hydrogen ions to pass back through the membrane, releasing that potential energy. And that's done by several different types of machinery, but the main one is our ATP synthase complex. And so just as most enzymes are named for the processes that they do, ATP synthase drives the synthesis of ATP molecules. So this chemiosmotic coupling is used by both chloroplasts in photosynthesis and by mitochondria in cellular respiration. And the main component of those are the electron transport systems and the ATP synthase complex. And so that's just a summary of how these two organelles are basically ATP producers for the cell. Now, some of the reactions along the way that we're going to talk about are oxidation reduction reactions, or redox reactions, that I mentioned at the end of our last lecture. So let's look at those a little bit, and they involve all these electron transfers. So that's actually really important in this process. Okay, so let's take a look at these oxidation reduction reactions. In short, we call that the redox reactions. Redox reactions are reactions that involve the giving up and the accepting of electrons. And when we think about this, ionic bonds come to mind very quickly. We talked about that in a previous lecture, but it can also be thought of in terms of polar covalent bonds as well. So when we take a look at our periodic table, those elements that tend to gain electrons very easily are going to be in this top right corner. And then those elements that give up electrons very easily tend to be on the left side of the periodic table, and the more ready an element is to give up electrons, the further down on the left they're going to be. So the trend tends to go in this direction for electrons. So let's take a look at our reaction where we combine sodium and chlorine atoms to make sodium chloride. So if we find our sodium element right here, you'll notice that it's in the first column, which means that it has one valence electron. Well, that's a fairly unstable situation, so it very readily gives up its electrons. Chlorine, on the other hand, is over here in the 7th column, which means it has 7 electrons in its valence shell. So it very quickly wants to grab somebody else's electron so that it can have 8 in its valence shell, completing the octet rule. So in our reaction then, our sodium atom here is going to give up an electron and become a positively charged sodium ion. And we call that part of the reaction the oxidation reaction. And so, easy way to remember that is over here for our oil rig. Oxidation is loss. And these statements are in reference to electrons. So oxidation is loss of an electron. The chlorine atom, on the other hand, accepts an extra electron and becomes electronegatively charged. And that is the reduction portion of our redox reaction. So reduction is gain of an electron. So these reactions occur all throughout the cell. Let's do one other example just so that you can get a handle on these. Okay, so let's step up our game just a little bit. So if we're looking at a redox reaction that involves an ionic bond, it's pretty easy to see where the electrons are being given up and where they're being hoarded. And so when we were looking in our previous reaction at sodium and chloride, our chloride was what we call the oxidizing agent. And all the oxidizing agent means is it is the one that comes in and steals the electrons. You can think of it that way. So oftentimes within a cell, oxygen is actually the oxidizing agent, and that's where that name actually came from. It doesn't have to be, but it oftentimes is. So if we look at the reaction that we have here, combining methane and oxygen to give us carbon dioxide and water, I could guess that if I gave you this reaction, you would say, I'm not really sure where the electrons are going. So how do we know then which part of the reaction is the reduction side, which is the oxidation side? How do we figure that out? So let's just start here with methane. So we have carbon and 4 hydrogen atoms. We know in this, based on our periodic table, that carbon is definitely going to be pulling the electrons closer to it than the hydrogen atoms are. But if we introduce oxygen to that, oxygen is even further over here on the right, and it likes to take away those electrons and that charge. So when we go from methane to carbon dioxide, note that we have gained oxygen and lost hydrogen. Well, a common thing that occurs in reactions is when you give up hydrogen atoms, you're also giving up electrons. So if we remember our mnemonic, oil, oxidation is loss. So when something becomes oxidized, it has lost or given up electrons. So our loss of hydrogen atoms here also indicates a loss of electrons. So methane has become oxidized into carbon dioxide. The other part of this reaction is that we had oxygen atoms here, and that would be a total of 4, right? And so when we go to the product side of our reaction, we note then that we have water. Well, now we have gained hydrogen atoms here and lost actually an oxygen atom. So as we gain that hydrogen atom, we have gained electrons as well. So the rig being the reduction is gain, so we have gained electrons as we have gained hydrogen atoms. So let's summarize that on our next page. I have a table that just goes ahead and tells you when we have all these various elements involved, what are you looking for when we're trying to figure out which part of the reaction is the reduction and which part is the oxidation. Okay, so when we're looking at redox reactions, and we know we have the oxidation part of that reaction and the reduction part of that reaction, it's easy to tell which is which when you are dealing with ionic bonds because it's pretty simple to see the gain or loss of electrons. When we go into other reactions, say that involve polar covalent bonds, for instance, sometimes it's harder to tell where you're losing and gaining electrons. So there are other hints. Particularly if we are looking at oxidation, we can have a gain of oxygen, a loss of hydrogen, or a loss of electrons, or some combination thereof. On the reduction side of the reaction, we can lose oxygen, gain hydrogen, or gain electrons, and again, some possible combination thereof. Not all redox reactions involve oxygen or hydrogen, so keep that in mind. Oxygen does tend to play a role in most of these reactions, which is why they got that name. However, that's not always the case. So one of the main redox reactions that we're going to be looking at here shortly is photosynthesis. So here is just a simplified version of the photosynthetic reaction. So we start with carbon dioxide and water, and using solar energy convert that to glucose and oxygen. So if we look at the beginning with our reactants, we have 6 carbon dioxide molecules, which in that means that we have 6 carbons and actually 12 oxygen molecules because of this 2 down here. And when that gets converted to glucose, we're going to notice that we have 6 carbons still, and our hydrogens now become 12, so we have gained hydrogen molecules, and our oxygen is now down to 6 instead of our original 12, so we have lost oxygen. And in that reaction, we have actually gained electrons as well. So that is the reduction side of photosynthesis. The oxidation side, we start looking at our water molecules. So here we would have 12 hydrogen and 6 oxygen molecules. And we are left then with 12 oxygen molecules. So we have gained oxygen on that portion, and we have lost hydrogen, and actually we've also lost electrons. So that is the oxidation side of photosynthesis. So photosynthesis is definitely a redox reaction. So why do we really care about these movements of electrons? Well, the movement of these electrons play a huge role in energy-related reactions in these pathways. So we'll take a look at photosynthesis in detail, and I think you'll see why these reactions are so important. So who can actually photosynthesize? This complicated process that we're going to learn in more detail, who actually does it? Well, I'm sure if I asked you that, most of you would say, oh, plants do, which is true. But plants usually refers to what you think of as like your houseplant or trees or anything like that. But in more general term is what we call an autotroph. Auto meaning self, troph meaning feeder, so it is a self-feeder. So it's an organism that's able to form nutritional substances from simple inorganic substances. And specifically for photosynthesis, that's using solar energy to complete that process, and those autotrophs are called photoautotrophs, photo being light. So this is when your Latin roots come in very easily. So those autotrophs, photoautotrophs specifically, actually do more than just photosynthesize, but at the very base of it, it is taking solar energy and carbon dioxide and water to then convert things into sugars, which can then also be used as building blocks within that organism. And then that organism creates all kinds of other things that we as humans use for a variety of things. So just examples here, food is the obvious one, and on this slide I've said food for heterotrophs. Hetero meaning outside of yourself. Heterotrophs would be those of us who ingest food. We obviously can't make our own food, so we eat plants and we eat animals, so those are heterotrophs. So our autotrophs produce food for the heterotrophs. Those autotrophs also, as a result specifically of photosynthesis, produce oxygen, which many organisms require for life because it's an important part of cellular respiration, which we'll get into later. And so that production of oxygen is crucial for life on Earth. The other part of oxygen production is that the autotrophs produce more than they need, and so it is released into the atmosphere, and for us that means a protective ozone layer that helps to keep UV rays at bay for us. Other things that autotrophs do, they produce material for clothing. I'm a Georgia girl, so cotton had to be in here somewhere. Building materials are produced, and then also some autotrophs have proteins that are used as thickening agents in toothpaste, in makeup, in ice cream, things like that. That's usually from our algae autotrophs. So those who can photosynthesize are what we call photoautotrophs, and they are really important, actually probably one of the most important organisms for life on Earth. So now that we know that our autotrophs, or specifically our photoautotrophs, are who can photosynthesize, let's take a look real closely at ones that we're familiar with. So let's look at flowering plants and look at our reaction for photosynthesis and where we get the reactants for those. So our photosynthetic reaction we know is going to require carbon dioxide and water, and that's just to start with. So in a typical plant, I have just a typical plant up here on the left. We have the roots that are below ground and the shoot portion of the plant, which is above ground. And most of us already know that plants use their root system to collect water. So water is going to be collected from the soil and brought up through the plant, and it's actually brought up through what we call vascular tissue. Again, in 112 we will go into detail about vascular tissue, but for now think of it almost like a straw that sucks up water and takes it to the rest of the plant that needs it. And so the places where we're going to accomplish photosynthesis are going to be those places where we can get solar energy the easiest. And so obviously that's not going to happen in the roots because that's below ground. So it needs to take place in those tissues that have pigments that are able to capture the sunlight. And so the pigment that is used most in photosynthesis is chlorophyll A and B, but there are accessory pigments as well that collect light too. But most of that is going to be in our green portions of the plant. We'll kind of go into that a little bit more as well. But suffice it to say that we need to take that water and bring it up to the green parts of the plant. And so the vascular tissue then will distribute that water to green portions. And one of the tissues that does most of the photosynthesizing is usually our leaves. And the reason for that is because of the second reactant that we need, which is carbon dioxide. So plant leaves are designed specifically with holes in them. And these specific holes are what we call stoma, or stomata is the plural of that. And where that stomata is, carbon dioxide will enter the leaves, the leaf tissue. And water actually goes outside through a process that we won't go into detail at the moment. So if we look at a cross-section of a leaf, you'll notice that there are these columnar-looking cells, and then there's a whole bunch of these other cells that just kind of look like round blobs. Well, those two layers are called mesophyll cells, and the mesophyll cells tend to have chloroplasts in them, and we'll go into exactly what chloroplast is, but suffice it to say right now that it contains chlorophyll, which can capture solar energy. And the nice thing is what we call spongy mesophyll cells, those are these right here that have a lot of space between them. They have all that space so that carbon dioxide will easily come in and diffuse throughout that space. If they were packed in super tight, then it would be hard for carbon dioxide to diffuse throughout the cells, and just based on our laws of diffusion that we already know, then the carbon dioxide wouldn't be able to come in as easily. But because the spongy mesophyll has that extra space between the cells, carbon dioxide comes in and diffuses throughout the leaf tissue. So now we know where we get carbon dioxide and water, and then of course I don't see that it's written in here, but you know that we need solar energy to accomplish photosynthesis. So we're going to talk a little bit more about how we capture that solar energy. So within those leaves of the plants, specifically in the mesophyll cells, the spongy and the palisade mesophyll cells, are chloroplasts. And remember when we were talking about eukaryotic cells and the differences between plant and animal cells, chloroplasts were one of the organelles that distinguished plant cells from animal cells. So chloroplasts are the organelles where photosynthesis takes place. So this image on the top right is just a layer of plant cells, and you can see all the chloroplasts shoved into these particular cells. So these cells are definitely going to be mass producers of oxygen, mass producers of sugars. So looking at the chloroplast itself, it has a double membrane. Remember we talked about how chloroplasts and mitochondria both have double membranes. And within the chloroplast, you'll see these stacks of what we call thylakoids. And the stacks themselves are called grana or granum singular, grana plural. And the thylakoids each have membranes with spaces in between them. They look like stacks of pancakes, really is what that looks like. And then there's what we call the stroma. And the stroma is just kind of that liquid part in between all of the stacks of thylakoids. So the carbon dioxide and water will diffuse into the chloroplast, so there's no active transport, they just naturally diffuse in. And the thylakoid membranes are where our chlorophyll A and chlorophyll B are embedded. So they're in the membranes of the thylakoid. And then in the stroma of our chloroplast is where the carbon dioxide is going to be reduced to carbohydrates. So that's the anatomy of the chloroplast and specifically where photosynthesis takes place. So when we look at the photosynthesis reaction here, it looks like a very simple reaction. But in fact, it is quite complicated and has multiple steps to it. And so a couple of things that we'll need to start thinking about is that photosynthesis requires enzymes, coenzymes, and you know what those are based on previous lectures. And then we've always said that it requires solar energy. Well, you know from things that we've talked about that sunlight itself doesn't really do much for us. So these plant cells have to find a way to convert solar energy to ATP, which is the energy currency of the cell, the way that things can work. So we're going to start with that process there. How do we convert solar energy into something that is useful in the cell? And so all of the things that we're going to be talking about from here on out are parts of photosynthesis. So photosynthesis has what we call two sets of reactions. There is the light reaction and the Calvin cycle. Those are not individual reactions, but those are groups of reactions that accomplish this goal of photosynthesis. So let's dive in and start with our variety of reactions that make up photosynthesis. So when we start the process of photosynthesis, we know that we need to capture that solar energy. So this begins the light reactions part of photosynthesis. And so put in a very simplified version, the light reactions are converting solar energy to chemical energy in the form of ATP and a molecule called NADPH. And so when we are looking at capturing that light, if you'll remember from previous science classes, that light travels in wavelengths. And if we look here at this table of various wavelengths, we have anything from really short wavelengths such as gamma rays up to long wavelengths such as radio waves. And what we mean by the wavelength is we would measure from the crest of one wave to the crest of another wave, and that distance between those two is the wavelength. And so visible light, the light that we can see, is only a small portion of these wavelengths. And so that falls between about 380 nanometers to 750 nanometers. And if you're talking about nanometers, that is 10 to the negative 9th meters, so they're very tiny. So when our plant needs to capture these wavelengths, it uses pigments, and these pigments are embedded in the thylakoid membranes. And specifically for photosynthesis, most of our typical green plants use what we call chlorophyll A, chlorophyll B, and carotenoids. These are the various pigment types. And these pigments only capture a certain range of wavelengths, and they do not capture others. So when we're looking at it, if we look here on the right, that is what we call the absorption spectrum of a particular pigment. So if we look at chlorophyll A and chlorophyll B, you'll see that there are kind of two spikes. One is in this section here, which goes from 380 to like mid 400s, and there's another spike over here in, let's say, the 650 to 700 range. And those represent blue light, and then down here a little bit of red light. Our carotenoids are also focused up here on the more blue end of the spectrum. What you will see that's missing is this section in the middle, and what that represents is green light. So somewhere in the 550 nanometer wavelength range, our pigments in our plants do not absorb that. So that's why when you look at a plant, it looks green to you, because green has not been absorbed. It is reflected back to your eyes, so you see green plants. So for each of these thylakoid membranes, these pigments kind of act like antennae. They grab the wavelength of light that they are able to absorb, and then they pass that along to what we call an electron receptor. And so each of these thylakoid membranes has what we call a photosystem, and the photosystem is a complex of pigments and an electron receptor, and all of that is in the thylakoid membrane. Within the thylakoid membrane are two photosystems, and remember we said photosystems consist of a pigment complex, such as chlorophyll A, chlorophyll B, and carotenoids, as well as an electron acceptor molecule. Okay, so there are two different photosystems within the thylakoid membrane, and they're just called photosystem 1 and photosystem 2. And they're named 1 and 2 just based on the times that they were discovered, not in the particular order that they are used within our pathways. So during the light reactions, electrons will follow a non-cyclic pathway for the most part. There are some that do follow a cyclic pathway, and we'll get to that in a minute, but most of them follow what we call a non-cyclic pathway. And it begins actually with photosystem 2. Keep in mind that was the one that was discovered second. So within photosystem 2, the pigment complex absorbs as much of the solar energy as it possibly can, and that energy is passed from one pigment to another until it fills up these pigments, and that energy is stored in what we call a reaction center, and you can see that right here. And that reaction center is actually a pair of chlorophyll A molecules. Now you can imagine as it's sunlight out and we have more and more energy being absorbed, that an electron occasionally gets so excited from this energy that it escapes the reaction center and gets absorbed by what we call an electron acceptor. And so this red arrow right here on the left really just demonstrates the amount of energy. So electron gets energized, escapes the reaction center, and is caught by the electron acceptor. Now, if we keep losing these electrons from our reaction center, then the photosystem 2 would eventually break down. So what happens is the water that we absorbed from our roots and is now up in the leaves and has diffused across into our chloroplast donates electrons. And so we have the breakdown of water. The electrons are donated to our photosystem 2, and we see then that oxygen is released. So this is where we have the byproduct of oxygen from photosynthesis. That's the product side, and that's the useful side for us as humans because we need to breathe that. When we've torn apart the water and we've donated the electron and we've released the oxygen, the remaining hydrogen ions stay within what we call the lumen, or the inside of the thylakoid. And that's going to contribute to a hydrogen ion gradient that we're going to talk about in a minute. Next, the electron acceptor passes the electron down what we call an electron transport chain. And this is just a series of electron acceptors, and every time we pass that along, we release a little bit of energy, and that energy is captured and stored in the form of a hydrogen ion gradient across the membrane. So when we have built up a hydrogen ion gradient, keep in mind that we have an enzyme within this membrane also called the ATP synthase complex, and just as the name implies, it synthesizes ATP. So it allows for that concentration gradient to be released, And because you're going from an area of high concentration to an area of low concentration, it's releasing that potential energy that we talked about, and the ATP synthase uses that to produce ATP. So already we've started to convert our solar energy to the chemical energy of ATP. This ATP is then used in what we call the Calvin cycle, which we'll talk about next, but let's stay here with our non-cyclic pathway of our light reaction. Our other photosystem, right here, photosystem 1, remember it is also capturing light, and those pigment complexes pass that energy onto a reaction center as well. And similar thing happens here. Once it gets energized so much, electrons escape and go to an electron acceptor. And again, we have a passing of the electrons down to, this time, a single acceptor, which is NAD+. NAD+ can actually absorb 2 electrons in the form of, along with hydrogen ions. So if you'll remember, when we accept hydrogen ions and accept electrons, we are reducing. So this is a reduction reaction. We have reduced NAD+ to NADPH. Both NADPH and ATP are going to be required in our next series of reactions, the Calvin cycle. And the Calvin cycle is where we start to produce those carbohydrates. But they're not required in equal amounts. In fact, the Calvin cycle requires more ATP than NADPH. So where do we get that extra ATP from? Well, sometimes as these electrons are being passed down their electron transport chain, sometimes they don't make it all the way and they come back over here, and we go through this whole cycle again and get rerouted and help to form that hydrogen ion concentration gradient, and which then goes through ATP synthase, producing more ATP. So that is the non-cyclic pathway of electrons. The cyclic part just being that those electrons can go back through that chain again, so that is a slight cyclical pathway there. So here's just another way to look at it. So as we are looking at this image, we've basically taken a cross-section of our thylakoid, and so we have the thylakoid membrane here, and so this is our thylakoid lumen. And then outside of the thylakoid, of course, is the stroma. So we have our photosystems 1 and 2 that are capturing light. We're harnessing that energy through our pigments. And then our electron transport chain. And as we have our electron transport chain, we are transporting hydrogen ions into the lumen of the thylakoid, and that is starting to build our concentration, and so increasing that potential energy. And of course here we have our NADP reductase. That's going to reduce NAD+ to NADH. And then our other enzyme, which is one of many, but one of the other enzymes, of course, is our ATP synthase, that is going to use this hydrogen ion concentration to drive the production of ATP from ADP and a phosphate group. And so there is our process that we talked about before. And so when we said chemiosmotic coupling, you can also just call that process chemiosmosis. Okay, I know that was a lot to take in, so we're going to stop that for now, let you have that sink in, review things. The light reactions are called such, by the way, because it requires light to be present for when those reactions are happening. The second part of photosynthesis doesn't require light to occur at that time, because it's converting the chemical energy that we produced in the light cycles, the light reactions, which are ATP and NADPH, and converting those to chemical energy in the form of carbohydrates. So we are going to discuss that part next, So for right now, go back and review the light reactions and redox reactions so that you can understand which parts require reduction, which parts require oxidation, and make sure you have that under your belt, and then we'll come back and do the Calvin cycle.